For today's lab, we're going to be using Mr. Potato Head and pipe cleaner chromosomes to show what happens in meiosis, and then to show genotypes and phenotypes. Genotypes are the actual genes that an individual has. And remember, we get those genes from our parents. So we get half our genes from mom, half our genes from dad. Those genes will then dictate the physical characteristics that we see in an individual, and that's the phenotype. So phenotype is that physical expression of the characteristics. For humans, what color hair do you have? What color eyes? What color skin? Are all controlled by our genes, but the phenotype, what we see, is that physical expression of those genes. When we look at genes, we have different variations of a gene, and that's what an allele is. It's a different variation for the same characteristic. I like to think of them as different flavors of a characteristic. So think of ice cream. You could have chocolate or strawberry or Moose Tracks or cookies and cream. They're all ice cream, but they're different flavors of that ice cream. That's what alleles are. They're different variations or flavors of that characteristic. So a gene for blue eyes or a gene for brown eyes. Different alleles for the same characteristic. Today we're going to be using dominant and recessive alleles in our lab. Dominant alleles are always given a capital letter. Recessive alleles are always given a lowercase letter. And the dominant allele will always get expressed over the recessive. So if we have two different alleles for a characteristic, and in our Mr. Potato Head characteristic, the D's are going to represent the nose. So the dominant allele is that Mr. Potato Head has a nose. And the recessive allele is that Potato Head does not have a nose. So with two different alleles, we can actually come up with three different genotypes. Potato Head can have two dominant alleles. Our potato can have two recessive alleles, or can have one of each. When they have two of the same alleles, we refer to that as homozygous. And if they have two different alleles, we refer to that as heterozygous. So we have two examples that are homozygous. This one with the dominant allele, and this one with the recessive allele. Therefore, we add another description after homozygous. This is homozygous dominant. This is homozygous recessive. We don't need to add anything here because heterozygous means two different alleles. So we have three different genotypes, but this one is going to code for the characteristic that it has a nose. This one is going to code for the characteristic no nose. In our heterozygous potato, because it has a dominant and a recessive allele, the dominant gets expressed over the recessive. And this potato would also have a nose. So we have 3 different genotypes, but we only have 2 different phenotypes. So today in lab, we're going to look at how meiosis distributes these genes and alleles into gametes, and how random fertilization with those gametes results in genetic variation in our baby potato spuds. So now we're going to go through activity 1 in your lab manual, which is on page 45. We're going to start with our chromosomes, and we're going to start with simple. We're going to have 4 total chromosomes in 2 pairs. A pair of blue chromosomes and a pair of red chromosomes. And for each of those chromosomes, we have alleles. In this one, we have the capital T, our dominant allele, and on this one, we have the lowercase t, or our recessive allele. So our potato got 1 chromosome from mom and 1 chromosome from dad. And now we have a pair of those chromosomes containing 2 alleles. And the same is true for our red chromosome. We have a capital E for our dominant allele and a lowercase e for our recessive allele. So if you're following along in your lab book, okay, question 1 says DNA replication in a living cell is very different from what we just did. What we're going to do before our chromosomes can undergo meiosis is they have to make a copy of themselves. So here's our dominant T, and when it makes a copy with itself, it has to be with another capital T. They have to be exact copies of themselves. Remember, when chromosomes replicate, they're held together by a centromere. So here is our replicated chromosome held together by that centromere. This chromosome has made a copy of itself. This chromosome will also make a copy of itself. And again, the copies are held together by our centromeres. Our red chromosomes are going to do the same thing. Lowercase e has to be with another lowercase e. So now we've made a copy of all 4 of our chromosomes. And let's look at a couple of questions that go along with that. The first question says DNA replication, which we've just done, in a living cell is very different from what we just did. How? Remember, the DNA molecule is actually a double-stranded molecule. And in order to make a copy of a chromosome, you have to break the bonds that hold those 2 strands of nucleotides together. Add new nucleotides to one side, add new nucleotides to the other. So it's a much more complicated process than what we did by just matching up our chromosomes. Now question 2 says, how many homologous pairs of chromosomes do we have? And the answer is, 2 pairs of homologous chromosomes. Because we have 2 red, 1 from mom, 1 from dad, and 2 blue, one from mom and one from dad. This is still considered one chromosome, even though it has its copy with it. They're not considered 2 separate chromosomes until we pull them apart in meiosis II. So what happens in meiosis with our chromosomes? In prophase 1, we're going to lose our nuclear membrane so that our mitotic spindles can attach to our chromosomes at the centromere and be able to pull them apart. Our chromosomes are going to condense down in size so that we can move them and pull them apart without them getting tangled. And then in metaphase 1, the chromosomes are going to line up with their pair. And so they're going to line up in the middle of our cell in 2 lines. The pair of blue chromosomes lines up and the pair of red chromosomes lines up. Because in anaphase 1, we are going to pull apart our homologous pairs of chromosomes. One of the red goes to one side of the cell, the other red goes to the opposite side. And the same is going to be true with our blue chromosomes. In telophase 1, those chromosomes reach the opposite ends of the cell. We get a new nuclear membrane back, and that cell divides in half via cytokinesis. At the end of meiosis 1, we have 2 daughter cells. And those daughter cells are haploid. They just have 1 blue and 1 red copy of their chromosome. They have half the number of chromosomes that we started with. This other daughter cell is the same way. It has 1 blue and 1 red copy of its chromosome. Notice in this example, this daughter cell got the 2 lowercase t's and the 2 uppercase e's. Because how those pairs of chromosomes line up in metaphase 1 is random. And by lining up in a random fashion, the daughter cells end up with some dominant alleles and some recessive alleles. Now, each of these daughter cells is going to undergo meiosis II. So in prophase 2, we again lose our nuclear membrane. We form our mitotic spindles, and our chromosomes are condensed. In metaphase 2, our chromosomes are going to line up in a single line because the purpose of meiosis II is to pull apart our replicated chromosomes, to pull apart our sister chromatids. So this cell lines up for meiosis II, and in anaphase 2, we pull apart the sister chromatids. One goes to one side, one goes to the other. And the same happens with our red chromosomes. This daughter cell over here will do the same thing. The chromosomes will line up in a single line and get pulled apart in anaphase 2. The result is we have 4 daughter cells. 1, 2, 3, 4. And each daughter cell is haploid. It has half the number of chromosomes. Our original parent cell started with 4 chromosomes, 2 blue and 2 red. Our daughter cells have 1 blue and 1 red chromosome. They have half the number we started with. But they have to have a blue and they have to have a red chromosome. When fertilization occurs, we get half the chromosomes from mom, half the chromosomes from dad. And in this simple example, if fertilization were to take place, we would be back to 4 total chromosomes. A pair of blue and a pair of red chromosomes. Back to our total number of 4. And one of the blue would have come from mom, and one of the blue would have come from dad. So meiosis occurs for one reason, to produce gametes, which are the sex cells, either the eggs or the sperm, which combine together during fertilization, that results in an offspring that's back to the correct number of chromosomes. Okay, now we're ready to start activity 2. And now we're going to have 5 different characteristics for our potato heads. And those characteristics are listed on page 48. Potato head is going to start as heterozygous for every one of those characteristics. So heterozygous means 2 different alleles. So capital T, lowercase t. Our E's are the same, our M's, our D's, and our A's all contain a dominant allele with the recessive allele. In order for us to undergo meiosis, we have to make a copy of every single one of those chromosomes. Just for time's sake, I'm just going to twist my chromosomes together. Our capital T's have made a copy. 2 capital T's connected together by the centromere. Same for our lowercase t's. And we will do the same for every one of our chromosomes. And remember, our homologous pairs of chromosomes are that we got one of the pair from mom and one of the pair from dad. And each one of these makes a copy before it enters into the steps of meiosis 1. And so remember, if it has a dominant allele, it has to make a copy and contain the dominant allele on the copy. So now our cell has made a copy of every single one of its chromosomes. Those chromosomes in meiosis 1 are going to line up with their pair at the middle or the equator of that cell. I'm going to use our lab tables here as our equator. It does not matter which side of that equator the dominant alleles are on or the recessive alleles are on. It's completely random how they line up at the equator. They just have to line up with their pair. So our 5 pairs of chromosomes have all lined up at the equator, and now we're going to pull apart the homologous pairs. And at the end of meiosis 1, we've created 2 daughter cells that are haploid, that have 1 copy of every chromosome we started with. Keep in mind, it still has its replicated chromosome with it. So each of these daughter cells has to undergo meiosis 2 in order to pull apart the sister chromatids. And in meiosis 2, our chromosomes all line up in single file. And during anaphase 2, we pull apart those replicated chromosomes. So this daughter cell is undergoing meiosis 2. And producing 2 daughter cells. And this daughter cell from meiosis 1 will also undergo meiosis 2 and pull apart its replicated chromosomes. And at the end of meiosis 2 in cytokinesis, we have produced 4 gametes. 1, 2, 3, 4. And each gamete is haploid. It has one copy of every one of those chromosomes we started with. One blue, one red, one green, one pink, and one white chromosome. And our gametes now are ready to be combined with another set of gametes from a different potato in order to simulate random fertilization. So what we have just done is we have taken one of the gametes that I produced in meiosis and one of the gametes that Bob Ford created when he underwent meiosis with his potato chromosomes. And now we have represented random fertilization. We now have 4 complete sets of chromosomes that are back in pairs. One of the pair came from the mother, one of the pair came from the father. And what we're going to do now is we're going to record the genotypes for these 4 Mr. Potato Heads. And then we will make our Mr. Potato Heads in the phenotype that's dictated by their genotype. Of our first generation of baby spuds. All of our potato heads started out looking like our original. Heterozygous for every characteristic. Therefore, the potato head exhibited all of the dominant characteristics or dominant phenotypes for each characteristic. And here are the 4 babies that we created. Baby A still has and exhibits all of those same phenotypes as our original. Baby B has no ears. Baby B was heterozygous recessive for the ear characteristic and ended up with 2 lowercase e's and has no ears. Baby C ended up being homozygous recessive for mouth type. Therefore, baby C is sticking out his tongue instead of smiling. Baby C was also homozygous recessive for arms. Therefore, he has no arms. And our baby D ended up being homozygous recessive for the nose characteristic and does not have a nose. And also ended up being homozygous recessive for a facial hair and therefore has no mustache. So all of our potato heads started off looking like this. And in our first generation, because of random alignment in meiosis 1 and because of random fertilization, we've gotten genetic variation in our first generation of potato spuds. We're now going to take 2 of these baby potato spuds and they are going to undergo meiosis again. They're going to produce gametes, we're going to have random fertilization, and we're going to generate an F2 or second generation of grandbaby potato spuds. have our F2 or second generation of potato heads. These are our grandbaby spuds. Potato head look like this. Here is baby W, here is baby X, here's baby Y, and here's baby Z. Now in your lab manuals, they will just be listed as your baby Y and Z. We have 4 of them from our original 2 Mr. Potato Heads that we started with. And as you can see, some of our Mr. Potato Heads now are homozygous recessive and therefore don't have ears or don't have arms. Some of them are homozygous dominant and will still exhibit the dominant phenotype. Some are also heterozygous, meaning that they have the one dominant and the one recessive allele, but they will still exhibit the dominant characteristics. So all of our original Mr. Potato Heads started as being heterozygous for each characteristic. We now have grandbaby spuds that are homozygous recessive, that are homozygous dominant, or are heterozygous for those characteristics. Nothing more than random alignment in meiosis 1 and random fertilization has created this variation in our offspring. Genetic variation in nature is essential for individuals to be able to survive in their environment. Okay, now we're going to analyze the data we collected with Mr. Potato Head. Our original potato heads all started as heterozygous for every characteristic. We're now going to see whether the data we collected support Gregor Mendel's ideas and patterns of inheritance, also referred to as Mendelian genetics. Gregor Mendel was a monk who lived in the mid-1800s, who knew nothing about chromosomes, knew nothing about genes and what they're made of, but recognized patterns in inheritance. He crossed pea plants together, thousands and thousands and thousands of crosses, to see what the offspring of those crosses contained in terms of physical characteristics. And he came out with ratios. Those ratios hold true as long as we start with 2 individuals that are heterozygous for our characteristic. So in our potato head, our dominant allele was for smiling, our recessive allele was for sticking out the tongue. Our original father, Mr. Potato Head, was heterozygous. The mother, Potato Head, was also heterozygous. We can now complete a Punnett square to figure out what the offspring should look like and what the probabilities are that they will be smiling or they will be sticking out their tongues. So remember, in meiosis 1, we pull apart the homologous pairs of chromosomes. This allele is on one chromosome, this allele is on another. They get separated. Half of the gametes here will have the dominant allele, half will have the recessive allele. And that is the same for our father, Mr. Potato Head. Half of his gametes will have the dominant allele, and half will have the recessive. Now we can set up our Punnett square. We can put the alleles the father can contribute to the offspring across the top of our Punnett square. And we can put the alleles that the mother can contribute down the side of our Punnett square. And now we can fill in our boxes. If this gamete was fertilized by this gamete, the resulting offspring would be heterozygous dominant. If we take this gamete combined with this one, we end up with an offspring that is heterozygous. If we take this gamete and this one, we end up with another individual that's heterozygous. We always put the dominant allele first. Makes it easier to determine the phenotype. And now these two gametes combine, and we have homozygous recessive. These are all the different genotype combinations that these two parents could produce in their offspring. Homozygous dominant, heterozygous, heterozygous, homozygous recessive. Now let's fill in their phenotype characteristics. This one has to be smiling. It only has the two dominant alleles. This one has the dominant and recessive allele and will express the dominant phenotype. So that individual is also smiling. Same for our other heterozygous. The last individual has two recessive alleles. Therefore, they can only be sticking out their tongue. You can only express the alleles that you have. And if you have two recessive alleles, you will have that recessive characteristic. The only way an individual can express a recessive phenotype is if they have the two recessive alleles. And now look, we have 4 possible combinations. 3 out of those 4 combinations gave us an offspring that was smiling. Only 1 out of 4 combinations gave us an offspring that was sticking out its tongue. This is a 3-to-1 ratio. In the offspring, there'll be 3 offspring exhibiting the dominant characteristic to every 1 that is exhibiting the recessive characteristic. A 3-to-1 ratio. This is one of the ratios that Mendel came up with in his patterns of inheritance. 1 characteristic, each parent heterozygous for that characteristic. 3 out of their 4 offspring will be smiling. 1 out of their 4 offspring will be sticking out their tongue. A 3-to-1 ratio. This is the ratio we would expect to see in the first baby spuds that we made. In other words, babies A, B, C, and D. And if we look at our results, we had 3 of those babies smiling and 1 sticking out its tongue. We did support Mendel's patterns of inheritance. We came up with a 3-to-1 ratio. What you have to understand is that we had a very limited small sample size. Normally we don't get that exact 3-to-1 ratio. So normally what we have to do is add data from previous years. And when we add data from other years, we come out with a larger data set. Statistics and ratios will more resemble, okay, the ratio we're looking for with a larger data set. And so, if I take these numbers and combine them with other years' numbers, we get 206 babies smiling, and we get 66 that are sticking out their tongue. Not a perfect 3-to-1 ratio, but pretty close. So with our example of 206 smiling and 66 sticking out their tongue, we had a total of 272 baby spuds. And if you take 272 times 0.75, or 75%, 3/4, you would get 204 that should have been smiley and 68 that should have been sticking out their tongue. We have 206 to 66. Very close to that precise 3-to-1 ratio that we were looking for. So the larger the sample size, the better it matches Mendelian's patterns of inheritance. Understand Mendel's patterns of inheritance and what we're going to be looking for in Table 7, we have to get a little bit more complicated. We're now going to look at 2 characteristics at the same time. And we're going to see what characteristics those offspring could have. In our example here, okay, here's our parent generation, homozygous dominant for each characteristic. The other individual, homozygous recessive. All of the offspring in the F1 or the first generation are all heterozygous for each characteristic. And so now we're going to take 2 individuals from the F1 generation, and they are going to produce our offspring. And when we look at 2 characteristics at a time, notice our Punnett square has increased in size. We now have 16 possible combinations. And when we look at those 16 possible combinations, round is a dominant characteristic, yellow is a dominant characteristic. And when we count up the offspring, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 out of those 16 offspring will exhibit both dominant characteristics. Round is a dominant characteristic, green is the recessive color. When we count up those possibilities, we have 1, 2, 3. 3 out of 16 offspring will have 1 dominant characteristic with a recessive characteristic. Wrinkled is our recessive characteristic here, along with yellow, our dominant characteristic. When we count up those possibilities, we have another 3 that show the other dominant characteristic with the recessive. And there's only 1 out of 16 possibilities that offspring will have 2 recessive characteristics. And in this case, wrinkled and green. In other words, we now have a 9 to 3 to 3 to 1 ratio. 9 out of 16 have the double dominant characteristics. 3 have 1 dominant, 1 recessive. 3 also have the other dominant with the recessive. And only 1 out of 16 has the double recessive characteristics. So let's look at how that applies to our Mr. Potato Heads. Table 7 in our lab manual is looking at 2 different characteristics, nose and ears. If our offspring have ears and nose, those are both the dominant characteristics. We would expect 9 out of 16 to have that combination. Here's ears, which is dominant, no nose, which is recessive. 3 out of our 16 offspring should exhibit that combination. No ears is recessive, a nose is dominant. Here's another 3 out of 16 that will have those combinations. In our last possibility, the double recessive characteristic, no ears and no nose. 1 out of 16 should exhibit that combination. So a 9 to 3 to 3 to 1 ratio. So with our baby potato spuds that we created, A, B, C, and D, here's our data. Baby A had an ears and nose. Baby B had no ears but a nose. Baby C also had ears and a nose. And baby D had ears but no nose. 2 to 1 to 1 to 0. The biggest problem with our data set is it's too small. We only had 4 individual babies in our data set. In order to have a 9 to 3 to 3 to 1 ratio, you need a minimum of 16 individuals. So we did not support Mendel's ratio in our data set because it was too small. So again, if we add data from previous semesters, we come up with totals of 112, 235, 231, 210. 112 have the 2 dominant characteristics. 35 have ears and no nose, 1 dominant with 1 recessive. 31 have no ears and a nose, 1 recessive, 1 dominant characteristic. In 10 of those exhibited no ears and no nose. It's not a perfect 9 to 3 to 3 to 1 ratio, but of that total that's down here, 112, 35, 31, and 10, we had a total of 188 babies. A true 9 out of 16 would have resulted in 106 in this category. 3 out of 16 should have resulted in 35 in each of these, and 1 out of 16 should have resulted in 12 here. It's not a perfect 9 to 3 to 3 to 1 ratio, but it is very, very close to that. The larger your sample size, the more it reflects Mendel's patterns of inheritance. And for this case, Mendel's patterns of inheritance relied on the fact that both parents were heterozygous for each of those characteristics to start with. And when that is the case, we see a 9 to 3 to 3 to 1 ratio in the offspring. If you look in your lab book, the question comes up, assuming these traits are inherited independently and based on your observations, how many of the offspring would you expect to show the dominant phenotype for both traits? There's our 9 out of 16. It goes on to ask how many of the offspring do you expect to have the dominant phenotype for one trait, but the recessive phenotype for the other. That's 3 out of 16 here and 3 out of 16 here. So we would expect in total 6 out of 16 of those offspring would have 1 dominant with 1 recessive characteristic. So we're going to fill out our last table in our lab manual, table 8. In this example, we are going to use our babies W, X, Y, and Z, our grandbabies. They were our F2 generation. Now remember, when we filled out table 7 and we analyzed table 7, we were looking for a 9 to 3 to 3 to 1 ratio. And we were looking for that ratio because each parent was heterozygous for our nose and our ears example in table 7. Table 8, we're going to look at nose and mustache. 2 other characteristics. But remember, babies W, X, Y, and Z came from our first baby spuds that we made. And when we look at baby W had a nose and mustache, baby X had a nose and mustache, baby Y had a nose and mustache. Baby Z had no nose and no mustache. So we have 3 with the double dominant, 0 in these 2 categories with 1 dominant, 1 recessive characteristic. We had 1 baby that had the double recessive characteristics. 3 to 0 to 0 to 1. Again, we don't have enough babies to even try to support a 9 to 3 to 3 to 1 ratio. So when we add data from other semesters, we don't have quite as much data because the lab got switched around a little bit, but we end up with 55, 26, 13, and 2. 55, 26, 13, and 2 does not even come close to a 9 to 3 to 3 to 1 ratio. Where did we go wrong? We're not supporting Mendel's patterns of inheritance with this table. The key comes in. Mendel's patterns of inheritance rely on both parents being heterozygous for each characteristic. Babies W, X, Y, and Z came from our babies A, B, C, and D. If you look back in the data tables, babies A, B, C, and D were not necessarily all heterozygous for each of those characteristics. So in this table, although we can look at the numbers that fall into each of those categories, we would not expect them to follow that true 9 to 3 to 3 to 1 ratio because we can't be sure that both parents were heterozygous for noses and mustaches to begin with. Hopefully this video has helped you to understand meiosis, patterns of inheritance, and genotypes influence the phenotypes or the physical characteristics that we see that get expressed.